triggering original fear memories. In upcoming clinical trials, Professor Merel Kindt at the University of Amsterdam, the Netherlands, plans to expose volunteers to fleet-footed spiders and tarantulas to provoke their fear memory. Professor Kindt is a clinical psychologist who began her research on modifying fear in 2008, inspired by earlier work with lab animals. That research convinced her that it was possible to trigger fear memories and destabilize them using certain drugs. It seems possible to target the fear memory itself so as to weaken the root of the anxiety disorder by weakening or even erasing the fear memory. Professor Merel Kindt, University of Amsterdam, the Netherlands. She does this by giving the beta-blocker drug propranolol in conjunction with the fear memory being triggered. Professor Kindt is using it in an entirely different way for phobias, by administering it after someone is exposed to a fear stimulus in order to interfere with the restabilization of their fear memory. Drugs that blocked protein synthesis were used to erase memories, but these are too toxic for people. By blocking them, the drug interferes with stabilization of the memory and dulls the strength of the memory and therefore the fear response underpinned by that memory. People will still recall that they were afraid of spiders, but the idea is that by weakening the memory we erase or weaken the bodily fear response next time the person encounters a spider, she explains. If you then observe a striking reduction in fear, this is not because the drug is still on board, said Professor. This is more difficult than spider phobias, because usually there are highly specific memories that underpin such complex traumas. To better understand how fear works, scientists are attempting to map out what fear looks like inside the brain, an extremely challenging task. But these brain images do not have the resolution to peer into the most ancient and mysterious parts that lie deep at the bottom of our brain. Professor Prof Gross, neurobiologist at the European Molecular Biology Laboratory, EMBL, in Rome, Italy. His core fear project sought to find which brain circuits were activated when mammals faced predators or stressful social situations, such as encounters with bullies. These experiments pointed to the epicenter for fear and anxiety as the amygdala, two almond-shaped structures found in the relatively recently evolved forebrain of mammals. We argue that the amygdala is just a gateway to real fear centers deep in the brain, said Professor Gross. The part of the amygdala that has been most studied for fear in the lab is irrelevant for fear of a predator or social threat. They had a conscious experience of fear and dread and a feeling like they were going to die, explained Professor. Human behavior is also complicated by our having a much more developed cerebral cortex, which makes up the largest part of our brains and allows us to suppress our fear responses. The EMBL lab in Rome recently released a preprint study showing that the mouse cortex can dampen inbuilt defensive behaviors of the rodent to threats. Our own highly developed cortex allows us to consciously control our own behaviors. Gross also recently reported on special hypothalamus cells that can map the spatial coordinates of where an animal encountered an intimidating rival. These cells fired whenever the animal returned to that spot, a social fear memory that encodes context and spatial memory. Such memories and fear of social defeats by rivals are likely to be important in territorial animals, which includes many rodents and also primates. Gross now plans to pursue these findings to fill in the many blanks in our understanding of how fear is stamped onto human brains.